skal vi over til at det jo ikke all sopp som vokser med trær. Arau her, som forsker ved Statens naturhistorisk museum i København, skal fortelle litt om sopp som vokser på insekter. Så vær så god.
I just could understand insect, and yeah. All right, so thank you very much for the invitation by Harvard and Mika and others that made this lovely invitation to come here. Well, I recently moved to Copenhagen a few weeks ago, actually, so I'm very new to the Europe Mycological Society and colleagues. I was the curator in mycology at New York Botanical Garden, and then now I moved to Natural History Museum of Denmark, and also assistant professor at the University of Copenhagen, and I work with cordyceps and associated fungi. As we all know, fungi are broadly undescribed and still unknown, so we know very little about these fascinating organisms. And the little we know is mostly about the decomposers, the mushrooms, and also the mycorrhizal associations because they have economic impact. And also a little bit about lichens. There is this beautiful symbiosis, and also the plant pathogens because they economic importance. But we know even less about the insect-associated fungi. So that's the topic I have been working the last 12 years or so. And just to give you an overview about all the groups of entomopathogens. So there are entomopathogenic fungi in basically all fungal major groups, although there's some unbalanced. Some groups are more diverse, others are less diverse. And this starts on the early fungal lineages, the aquatic or the so-called chytrids. And they have these really interesting life cycles that they have different hosts, and they do some amazing stuff. For example, they infect the copepod here, and then they can infect also mosquito larvae, or kill the mosquito larvae and then come back to the copepod, or it can remain inside the mosquito larvae, as we can see here, as these cells, and then they can remain there until the mosquito reaches the adult phase, and the mosquito will lay fungal sporangia rather than eggs. So that's a really beautiful strategy because they will remain in the environment that there are other mosquito larvae, right? So it's a really, really interesting strategy. Another group that also are called the zombie fungi, but although not related to Ophiocordyceps, a different phylum, far related, but they also do these very interesting things where they fill the body of the insect and making the dead insect to spread the wings like that. So it's like an invitation for others to come and try to mate. But when they come and touch them, they become infected. So it's another beautiful strategy to transmit spores. And these also are particularly interesting in Massospora and Strongwellsii because they sporulate from the living body of the hosts. So usually insect parasites, they need to kill the host in order to produce the spores and sporulate. So they, from parasitic, they switch to a saprophytic. And then they are yeast-like symbionts, and then they switch to a hyphal and filamentous phase. But this one sporulates from the living body of the host, which is quite unusual. And within the Basidiomycota, surprisingly, there are very, very few lineages that evolve the ability to parasitize insects. And those are this one here, Fibula rhizoctonia, which are these orange sclerotia here, and the white ones are termite eggs. So what these fungi do is that they mimic the egg of termites. So when termites are out foraging, when they find these structures, these fungal structures, they think those are eggs, and they bring them inside the nest. And they switch from a saprophytic, free living stage, and then they switch to a parasitic stage once they are inside the termite nest. So it's also a beautiful strategy, I think. And Septobasidium, which is, this is a section, And this is a tree, and that's a scale insect, and that's the stylet sucking the sap, and that's the fungal filaments that grows on it. So it penetrates the insect and uses the insect as a gateway into the phloem. So it doesn't kill the insect in this case, but just makes them slaves, basically then. And we have my favorite group, which is within the ascomycetes, the hypocrealian fungi. It's a very diverse order of fungi. And they evolved back in the Cretaceous, around 150, 130 million years ago, when the flowering plants were also evolving. So we have the radiation of angiosperms and all these new species and new lineages, which also means new opportunities to be exploited by insects. So together with the radiation of angiosperms, we have the coevolution with the insects. So the insects were also exploring these newly evolving niches of the angiosperm, which are flowers, fruits, and all these new resources that were emerging back then. And as we can expect, fungi also follow the trend and co-evolved with the insects that were co-evolving with the plants. So if we compare the phylogeny of these three groups, the plants, the insects, and entomopathogenic fungi, and these green bars are this period, this Cretaceous, and we can see radiation starting at these stages concurrently. So we can parsimoniously suggest that this is a co-evolving series of events that happened. And this is a round phylogeny of the order Hypocrealis. So the message here is, well, each branch is coded by different colors, as you can see, and each color means one type of host association. So green, here, the basal lineages are broadly associated with plants. They are plant parasites. Whereas the more derived ones here are broadly associated with insects. So those are the groups that have been studied as major three families. So, but as we see that they are very plastic, so they do what we call host jumps. So they are originally plant-associated fungi that evolved the ability to infect other fungi and become mycoparasites, and also infect all sorts of insects, and also sometimes they come back to plant, as we have here a group in Clavicipitaceae. So now we're gonna just focusing on the major three families just to give you an idea. So the colors here means the different types of hosts. For example, this pink are fungi-associated mycoparasitic lineages. Purple are the spider parasites, and then we have the yellow infecting Lepidoptera and also other groups. So we see that the host association is very diverse across this group. And then we have Clavicipitaceae that also, in fact, they switched back to plants. So this is broadly associated with plants, plant endophytes or plant parasites, and also other types of insects. And then we have the family that include the zombie-ant fungi, which is Ophiocordicipitaceae. Here we see in red the lineages infecting ants, and in blue the ones infecting beetles, and so on. So I will come back to that and tell the story about their evolution. But just to give you an idea how diverse, ecologically diverse these fungi are. Which includes, also I just mentioned, the zombie-ant fungi, which is what I will be focusing today. And as the zombie-ant fungi, naturally they infect ants, which is a very broadly distributed group of organisms and very abundant, and they comprises a huge biomass in the forest. So meaning that this is an amazing resource for the fungi to explore because they are so abundant, so they're everywhere. And why is it a good model system to study ants and parasites? So there's here a few reasons. Because we know very well the biology and the evolution and the systematics of the ants, and that helps us to create hypotheses on the coevolution of these fungi and the ants, because we know the biology of the ants very well, and not so well the fungi. But if we have half of the story already already established, is better for us to estimate things about the fungi. So that's a typical zombie ant when they are infected. So they have this biting behavior that they seek for a summit position. I will show a short video that will represent better the life cycle. But in quickly showing, so they bite and then they grow these fruiting bodies that, depending on the environment, the forest can take in the Amazon a few weeks, or in temperate zones can take a whole year to develop. I will touch on that too. And field work, so that's, I like to include this slide because I have done work in the temperate systems. And that's one of the most important parts of the work because these are really small fungi usually. So it requires some eye training to find them. And now I'm interested to investigate these fungi here in Arctic systems because now I'm based here, so it's I really want to investigate more these fungi. They are here, so I visited the herbarium yesterday, the Fungarium, and I could see some really interesting, even new species there that hasn't been described from here. And well, here's 2 photos I took yesterday very quickly. So here's a very interesting, looks like an assassin bug or something with Ophiocordyceps growing out of them. And this is more common, and there are some records in Copenhagen even that people found many of those in parks. So I'm really looking forward for the summer season so I can go myself and try to collect them and establish some local system here, which is this beautiful thing. And something that's really intriguing that these fungi, this is the same species, but these are larvae and this is an adult beetle. But that's Ophiocordyceps entomorrhiza. And they're very often, they display these white fungi, which some people consider as the anamorph of the Ophiocordyceps, and it's called Thylacidiopsis nigra. But it also resembles a lot mycoparasites, like polycephalomyces, for example. So the first low-hanging fruit to investigate a local system would be to find out if this is a mycoparasite that's often associated with that, or if there is actually an anamorph or an asexual state of Ophiocordyceps. So that's the first thing I would like to investigate here. And then back to the zombie ants. So we have back then, when I started my PhD, so the zombie ants were represented by a single branch in the phylogeny. So Ophiocordyceps unilateralis, or everything would be included in the pile of Ophiocordyceps unilateralis. Until we started to look at it more carefully, and then we found out that this is actually a huge complex of species. There could be, we use molecular, but they could be really easily differentiated based on morphological features. It's really clear. The spores are very different. The germination is different. The anamorphs are often different as well. And the host association is different. So from one branch here, we now publish a few papers on that. And now we have a much better resolution. So from one branch, now we have 36 species described. And even in a different clade here, we found also species that manipulate the host behavior. So that occurred multiple times along the evolution of Ophiocordyceps. Not just unilateralis, but there are other species too. Yeah, this is a species we described that was just featured in the documentary. Yeah, and many people ask me, how many species of zombie ants? So there were 1 species, and now it's 36, but how many we should expect? So this is showing us some preliminary data, and we found basically that half of the carpenter ants, the Camponotus, in the Amazon are infected by zombie ant fungi. This is very conservative. I know it's more. I have unpublished data for way more species than that, but let's be conservative here and consider that half of the carpenter ants are infected by Ophiocordyceps. So here's the Camponotus, the carpenter ant, and that the gray bar means the number of species of ant, and the black bar number of species of Ophiocordyceps. And we can see that half of the Camponotus are infected, but other genera, like Pheidole here, there are 72 species in the Amazon, were never been found infected by Ophiocordyceps. So that's another interesting research question. Why some groups act as magnets for fungal infections, while others are completely apparently free of infection. So, but if we consider the rate we found in the Amazon, that half of the carpenter ants are infected by fungi, and we extrapolate to the global diversity of carpenter ants, which is 1,200, more or less, species. So we end up with estimations of 600 species. And we described about 30, so we still have 570 species of fungi to describe just on carpenter ants, just in the genus Camponotus. Not considering Polyrhachis or other Camponotini ants in Africa and Oceania that I'm not even mentioning here. So the diversity is huge, huge, incredibly amazing. So there are lots of PhD theses to be developed just in the taxonomy of this particular group. And now this is that's one of the reasons that these fungi became more popular, is because their ability to manipulate the host behavior. Right? So there's a term called extended phenotype that was coined by Richard Dawkins, which is the genes of the fungi that has been expressed in the phenotype or the behavior of the ant. So the genes of the fungi are making the ants to leave the nest and bite onto these summit positions. So that's what we call extended phenotype. And then a few years ago, we published a hypothesis on how this might have evolved. So which evolutionary forces were driving this behavior manipulation. So to do that, we reconstructed the phylogeny of Ophiocordyceps, and we again color-coded according to the host association. So we have this clade here, the zombie ants, on red. So red means infecting ants, and blue, the blue ones here, means infecting beetle larvae. So the beetle larvae we found nowadays, they are often buried or in logs in the Amazon, and they occupy that niche. And they're also a niche that's occupied by the carpenter ants. They nest there, so which means that they overlap the niche. They occupy the same location in the forest. So that creates an opportunity for the fungus to jump. Remember the host jump I was talking about? So here is the same. So at this place here, in the number 2, so the ancestral that were in our results suggest that were infecting beetle larvae, as the basal lineages here still do. So in the number 2, there was a host jump from the beetle to the larvae. I'm sorry, the beetle larvae to the ants, because of the niche overlap. But imagine a beetle larva that is a naive, solitary, fat, and thin-skinned. So it's really an amazing resource to be more easily infected, and also has a lot of energy to be exploited and form these beautiful fruiting bodies here that's very, very abundant. But when they jumped from this naive, solitary, unprotected beetle, they found something in the ant colony that's called social immunity, which is the public health systems of the ant, basically. So when the other ants found that one of the colony members are infected by fungi, they try to rip them apart and put in these trash chambers or kick them out from the nest, so they won't transmit to the other nest mates and therefore collapse the nest. So that's called social immunity. So our hypothesis was that once this ancestral fungi jumped from beetle larvae to the ants, they found this social immunity, so they couldn't develop in time. So the ants would recognize the infected ants and would stop the fungal growth, because they rip the ants apart and the fungus die eventually. So the strategy that the fungus found was to be undercovered, and then right before the other ants could recognize as the infected ants and the ants display the symptoms, it will control the behavior of the ants in the brain in the ways that we still don't know how, and the ants will leave the colony, and therefore avoiding the social immunity. So that's the hypothesis, that the social immunity of the ants was a driving force that created this ability to manipulate the host behavior. And here, one of my favorite examples. So this behavior manipulation is not uniform. They're just infecting the ants on the ground and make them climb. There are some examples that are different. This is one of those. So this ant, Monassis bispinosus, it's an Amazonian ant, and they nest on the canopy, not on the ground like the carpenter ants. So they nest on the canopy. And when the ants are infected, they descend from the canopy and die on the base of these trees. They're often covered by moss, Octoblepharon albidum more specifically. And if we look at this carpet of moss, and we see that this is sporophytes here, right? But look at the fruiting body of the fungus. That's really, really similar to the sporophyte of the moss. So the fungus is mimicking the sporophyte of the moss. So the fungus is controlling the behavior of the host and making them to die in a particular location that the sporophyte of the moss is nearly identical to the fruiting body of the fungus. So that's really something that we still need to understand why they are doing that, why they drive the host to die onto that particular location in the forest. But this by itself, I think, is really, really impressive strategy. And this shows that there is also, this is not, as I said, is not uniform, and this shows that there is variation across these behavior manipulator ants. And this, we wanted to understand the differences, because if they bite onto leaves, they wouldn't be able to develop in temperate systems, because the leaves would fall on the ground and then on the fall season, and they are very sensitive fungi. So if they are removed from the location that they made the ant to bite, it will stop growing, it will not grow. So we found that in temperate systems, they bite onto twigs instead. So in the tropical forest, they bite onto leaves. In the temperate systems, they bite onto twigs. And here is just to show how they grow fast. So that's the day one, we found the ant biting, and just 2 days later, there was a stalk already developed. So they grow really fast and also produce conidia already at this stage. But if we compare with the temperate system that is here, so we see that day one is mid-July, end of July. So we will just mature here over 300 days after, because it will bite in the summer, and then will take a while until the fungus develop. And then the winter comes and literally froze the ant with the fungus, and it survived freezing times. So it has to bite onto twigs, so there was a local adaptation to these cold environments. And we asked the question, who came first? We expected that the leaf in the tropics came first, and then they were migrating and gaining latitudinal gradients into more colder climates. They were adapting. So we could see here that the ancestral in green means biting leaf, brown and blue means biting twigs. So we can see that these evolved at least 4 times independently here. So there's one species in Japan that is cold, and also one species in South Carolina that is also cold, and they both developed this strategy to fine-tune the biting behavior in order to make them not bite leaves, but biting twig instead. And if we plot these records in the map, we can clearly see this pattern, that in the tropics, again, green means biting leaf, and brown means biting twigs. And in the tropics, they bite leaves. And here in the United States and colder places, they bite onto twigs. Here in Japan, you see that they bite leaves too, but this is an evergreen forest, so it doesn't matter. They can bite onto leaves because the leaf will not fall on the ground during the fall season. That's a strategy they didn't need to change because the leaves will be there. So, and then we found that this was a local adaptation to colder climates to fine-tune the ants to bite onto twigs instead. So now I will just quickly mention some of my future plans to work in Copenhagen and on new systems. So as I showed that these Ophiocordyceps are very diverse, and not just Ophiocordyceps, but Hippocrealian fungi are very diverse, and they infect a broad range of hosts, and there's lots of different species complex throughout these this order and across these families. So I still want to explore the zombie ants and the mycoparasites that are associated with them, the gibellulas infecting spiders that's understudied, the acanthamyces on moths, And then we have the hypocreloids, Aspersonia, infecting scale insects that are economically important, and the beetle larvae. Those are all complexes of species that haven't been studied. So there's a cryptic diverse behind those ones, which also include the mycoparasites. So we have the ants being infected by Ophiocordyceps. And then we found out there are also fungi infecting the Ophiocordyceps. So we have the ant, Ophiocordyceps, and another fungus infecting Ophiocordyceps. So this is a case where we discovered 2 new genera, Niveomyces and Torrubiellomyces, infecting one single species in Florida, Ophiocordyceps campanoti floridani. We described in 2018. And then 40 years later, we described these 2 new genera just associated with one species. So imagine if we look into those 36 described and potentially 600, if each of those harbor their one mycoparasitic species, how many species there are still to be discovered? So it's a huge diversity of entomopathogens and mycoparasites that are still to be discovered. And this is a good example on that. And there's also, I didn't include here, but we more recently published a study based on a Thai system. So in Thailand, there's also these mycoparasites that are really specific. One group of entomopathogen will have its own mycoparasitic species infecting them. Not just in the unilateralis, but Ophiocordyceps dipterigena or Gibellulas infecting spiders. All of those also have mycoparasites associated with them. And that is becoming gradually my favorite group. So we have Ophiocordyceps, as I mentioned, infecting 13 orders of insects, including Hemiptera, the sap-sucking insects, which include cicadas, plant hoppers, tree hoppers, aphids, scale insects, all those. And you see that the phylogeny here has these white names and red names. So the white names means the classic entomopathogen, the fruiting body forming species. As we can see there, there's a cicada nymph and this long fruiting body erupting from its body. And then we have these that we are working on that to publish soon, hopefully, that are cryptic lineages that lives inside these hemipteran insects that do not produce fruiting body, but they are beneficial to their hosts, which is called endosymbionts. So those are yeast-like stages that don't switch to the filamental stage. So they will remain unicellular or in small groups, small clusters like that. And this is a technique called FISH, fluorescence in situ hybridization, that we can design some, prime some molecules that will bind to the fungus and will make them glow. So we can see them better in a confocal microscope. So here we see the body of the insect, and we see in pink the bacterial symbionts, and in yellowish here we see the yeast-like cells. They're very abundant. So this story is interesting because the cicadas, they usually have 2 main endosymbionts, Suscia and Hodgkinia. So when my colleague first found that in 2018. So there was just one of those symbionts. The other one was not there. But he was, what's wrong here? Because they have to have both. They're essential endosymbionts that synthesize essential amino acids. And then they found that the bacteria was missing, the Ophiocordyceps was there. So what happened here is that the Ophiocordyceps colonized the hemipteran insects and kicked out the bacteria. So there was a replacement of the bacteria. And the fungus has, in this case, very reduced genomes, and they lost the ability to produce the fruiting body. And it's good for the fungus because they don't need to produce the fruiting body, shoot spores into the environment, and hope to find a new host. But those are transmitted by the eggs, so they're transmitted vertically. So that ensures that 100% of the offspring will be infected or colonized by these endosymbionts. And we also found out that those are, each of the Hemiptera, what's going on? The Hemipteran groups, they are, each of those origins of these red clades, the endosymbionts, is driven by one host group. So one group, they are the first origin, they are associated with exclusively with scale insects. So we see that there is a convergent evolution that occurred multiple times along the evolution of Ophiocordyceps. And this is just the tip of the iceberg. We know nothing about them. So it's really understudied. So we hope to really broaden this phylogenetic tree and also the association with the host. This might have been throughout all the insect groups. We just don't know. We never looked, actually. But the interesting thing here is that the diversification of the fungi are driven by one insect group. So insects are promoting fungal diversities and act as islands of biodiversity for these insects. But those are cryptic, so we're just finding out more recently. And something that I have been more interested more recently, this paper is currently being reviewed. And those are fossil Ophiocordyceps. So there's a genus that was created back in 2008, is Paleo-Ophiocordyceps, which is interesting because they have features of two clades in Ophiocordyceps. So we could suggest that these are the ancestral of these two clades because they have features of both clades. And then we are proposing these two new species, one infecting a fly, which is here, and we can see the club-shaped structures, which is analogous to extant species infecting flies in South America and here in Ethiopia. And then there is this beautiful club-shaped structure coming out from the back of the head of the fly. And this is in amber. This is about 100 million years old. And this is one infecting a larva, the egg of ant, an ancestral ant, like 100 million years ago as well. So and then we did a CT scan, and then we could see these perithecia were present there. There's insects don't have such a structure, and this really we could even see the cells here producing conidia at the tip, like we still have the Irsotella that produce that in the extant species. So that's another thing that I'm very interested. And the fossils are interesting because we can use those to calibrate the phylogenies, and then we can hypothesize when these lineages evolved. So as I said, those species we show they have characteristic of both clades here. So we have the Sphicocephala clade, and then we have the Irsotella clade. So those ancestral species have morphological features that common to this clade and then to this clade. So we can suggest that the ancestor was here, right? And then we can hypothesize that they evolved 200 million years ago. So we can use these fossil calibrations to infer when each of those clades and their associations emerged 200 million years ago. And that's something that in Copenhagen we have a huge collection of amber, and I'm looking forward to take a look in some of those, and maybe we can have something hidden there that nobody found it before. And this is not just interesting from the scientific perspective. So I think this is a really fascinating group to spread the word about mycology. So all of us here are already convinced that fungi are amazing, they are fantastic organisms, but we still need to convince the world out there. And I think those fungi are really good examples to caught the attention of people and then make mycology more widespread and more known and attract younger generations to get interested on that. And that has been more recently been featured in documentaries. I didn't include a photo of that, but the TV show The Last of Us, that is all based on Ophiocordyceps and these zombie ant fungi. So that was the inspiration of these guys that made the TV show. And that has brought a lot of attention to this group and to cordyceps and these manipulative fungi. So I think they are also really great tools to communicate science, communicate mycology, and the importance of fungi. And now I would like to thank you, Howard, for the invitation to bring me here. Mika, I don't see her. Oh, yeah. that showed me the Natural History Museum collections yesterday, and was really nice, and we hope this is just my first visit and the beginning of a long-term collaboration with colleagues in Norway. So thank you a lot.
Well, it was an extremely nice presentation on an exciting topic. As you look forward to your studies into the boreal temperate biotopes, do you expect that because of the less optimal conditions for rapid growth and fruit body development, that you will find a large diversity of species that have evolved alternative ways of dispersal, like conidia or through the eggs, or that is as of yet undiscovered?
Yeah, that's a really interesting question. So as I said, we know very little about these fungi. We're just starting to understand their biology. There was a paper published 3 years ago showing that Ophiuchordyceps sinensis, the very famous medically important fungi, especially in China and Asia, that they are transmitted trophically, meaning that when a caterpillar eats well, let me come back. So this study found that the spore, the fruiting body that is produced in the caterpillar, the classic entomopathogen type of thing, and they found that this fungi has also an endophytic stage, so they live inside plants, protected. So when the caterpillar eats the roots of these plants, when they chew that, this endophytic stage will get into the insect, and then they switch, they wake up, and I have the potential to be a parasite. And then they start to consume the caterpillar, and then grow the fruiting body, and theoretically, they couldn't prove that in the paper, the spore will land on the leaf or on the plant, and then will penetrate and will come back to the endophytic stage. So perhaps the endophytic stage is the dormant, or the stage that they survive the winter. Or another hypothesis is that they can survive and remain inside the host body throughout their lives. So there's something, maybe when they change from the pupa to the adult, or from the caterpillar to pupa, so that might trigger the pathogenicity of these fungi, and they might be living there, and then eventually they become an entomopathogen. So maybe they are just hidden and living their lives inside the host body, and something triggers them to be a parasite. So therefore, two hypotheses: one, that they can survive inside the host body for a long term, and then eventually become a parasite, or that they can be endophytes that can potentially infect insects. That could be the case for colder climates like here, but we still need to study that. That's a really interesting side of the story.
Thank you. You can take it.